So, hello everyone. Those of you who are joining for the first time, this is Maushandra. I'm the TA who is going to help you with the problems for this course. All right. So, a brief introduction. I'm actually a PhD scholar at the Department of Chemical Engineering, IIT Kanpur, and I'm working in the area of tissue engineering. And I have, through my research, I have gained experience in various biofabrication techniques. Few of those we have already discussed during the course, so you're already familiar. Um, the If you want to know more about my work and all, I have a website that you guys are free to look at. And also queries, like I really like if you guys have some queries and ask. It helps me learn that you are able to understand, and if and when you're not able to understand, so queries is a good thing. Is the part of learning, and it helps me realize that you're able to learn. Okay, so we have been meeting like this every Saturday. Previously, the time was 4 to 6, but from this week onwards, and for the remaining week, the time will be 6 to 8 p.m. Please note the changing time. The time of the session will be from 6 to 8 p.m. It is solely because of the reason that I have moved to United States for an exchange program, and to fit my schedule here, that's why I have changed the timing. I hope that's not a very big change from your side. Even if you're not able to attend the session, the recordings will be available for you to watch later. The meeting link and everything remains the same. The notes will be available to you. Recordings will be available to you. And yes, I have been taking note of how many people are attending every session, and that can also be seen in the sheet that you guys have. Although I was planning previously to, keep an extra session towards the end, till last week, I was actually very much determined to do that. But due to my this change schedule, I will not be able to do that. Okay, so our last class will be on 14th September to discuss the last week's session, and after that we won't be meeting. So if you have any queries, I request you to ask during these sessions. And I don't mind if the query is not from the week's discussion. It can be from any topic which is taught prior, and I'll be happy to help you out. Okay, but I hope that you are like studying as well. If you don't study, then okay, there is no point in asking for queries. Okay, so yeah, so the platform remains the same. It is MS Teams. The chat option is not available. You have to unmute yourself and ask queries, but the hand raise option is available in case you want to, like raise your hand and you want me to call upon you or something like that. But you're feel free to, um, just unmute and ask. So briefly, what is taught in week 6 content, like what was taught during the week 6 lectures. So I decided to go 2 lectures behind, like starting from week 5, because that is when we started the topic of tissue dynamics. Okay, and I'll be in so today's questions will be started with the topic tissue dynamics, and then slowly we will solve some questions on several of eight processes, and the small topics that were taught, that is how to deliver cell signaling molecules for tissue engineering applications, and what kind of bioreactors etc are used, like water bioreactors and all. So that will be discussed very briefly. Okay, so the main focus for this session will be on the tissue dynamics and the several fate processes that control the tissue dynamics. So what is tissue dynamics, or what do you understand by this term? So that our body is always changing, right? As we grow old, our skin changes, our hair changes, right? Everything changes. And that change is visible to you in the mirror, but also in our body. The change is going on, like even if you don't know. Every tissue is at every point of time is either repairing itself or trying to do the processes to maintain its steady state function. And if someone is pregnant, then in their body the developmental process is also happening to give rise to a new life. So the whole our whole system is very dynamic in nature. It is not a static thing. Like if the god has made you like this, you're not going to stay like that forever. That is what I mean. Okay, so these processes that people have studied through research are the tissue homeostasis, which means our tissue at any point of time is trying to maintain its steady state function. So what is steady state? What this term means? It means that your the tissue is functioning or showing the same function irrespective of time. Okay, that is steady state. You must have used this term a lot when you have done some problems in physics, right? Like develop some mathematical models. So it is easier to assume in those scenarios that there is a steady state, means that that thing, that function, or it does not change with time. So every tissue has that a steady state function, and it is trying to do its best to keep it like it. Okay, so let's say the function of skin. The main function of skin is to act as a barrier so that our internal organs are safe. So our skin, you won't see that any part of your skin is missing at any point of time, right? Any part in from any part of your body. Until and unless some injury has happened, our skin is intact as it is. So that is a homeostatic state. It is trying to maintain its barrier function. Okay, similarly, your brain, your heart, every organ or every tissue has a steady state function that you have, where we are very much familiar with. That is what we are taught when we are introduced anatomy. So that is homeostasis. Now, repair. So, in case there is some event that took place and your homeostatic state is compromised, so that is when we say that the tissue or organ is injured or diseased, right? So, in those cases, another dynamic state is very important or comes into picture. That is the tissue repair functions. The tissue repair process starts happening. So that is another dynamic state. It is conditional. Okay, it is not like homeostasis. It is always there. It is persistent. Tissue repair will only happen when you're when the sanctity of the tissue or the organ is compromised. And the last dynamic state is tissue development. This happens when uh, a new life starts forming. Okay, during the process of reproduction. So this is also for any person happens only once inside his or her mother's womb. That's it. So our from zygote we become this multicellular organism. How it is happening? So that is happening through tissue development process. Okay, so I hope briefly I could make you recall what are the tissue dynamic processes. Now all of these processes are regulated by cellular fate processes. And if these cellular fate processes won't happen, then you won't be able to see the dynamic state of a tissue. So at any point of time, to maintain the dynamic state, the cells in that tissue or organ are adhering to a surface. If it is an adherent cell type, it will migrate, it will show the tendency to divide. If there are stem cells involved, then they will differentiate. And if the cell has grown old or no longer needed and all, so it will undergo cell death or cell apoptosis. Okay, so these are five main cellular fate processes. Okay, and We will means these terms, I think you must be already familiar when you have studied them during your cell biology course. But in this course, we are mainly focused from the perspective of a tissue engineer, how a tissue engineer sees these processes, how they like characterize these processes when they are trying to prove the success of their tissue engineered model or a graft. Okay, so from that perspective, we will study these processes. And lastly, so, we have already studied the role of the biomaterials, which act as the artificial ECM, and the cells that interact with this ECM and creates the artificial tissue. But in order to uh, control the behavior of this artificial tissue, we supply some growth factors or cytokines or chemokines. So these signaling molecules, these are called basically signaling molecules. What they does, they like control the growth of the cells or the control the cellular behavior on your artificial tissue. And this is very important when your target is to create a functional tissue for a particular application. You cannot expect that the cell and the biomaterial will just come together and give you what you need. Um, it is not always true. When your desires are more complicated, when your application is more complicated, and when you're trying to mimic what is there in the body, it is not what it is. In the body, there are always these small molecules, small signaling molecules that are trying to interact with the cells and tell them what to do, how to behave, okay, where to migrate. So these things are always happening in your body. And for tissue engineers, we are interested in mimicking those behaviors inside our body. That is why we are interested in knowing the ways to deliver this cell signaling molecules in our artificial tissue. Okay. Now, when it comes to culturing these tissues, artificial tissues, in vitro means outside the body, then um, the traditional methods may not work always or may not give you the correct desired results. But traditional methods, I mean the traditional cell culture methods, where the flow is static, there is no flow, okay? Um, or there is no, what do you say, volume, sufficient volume of liquid available, or there is no sufficient exchange of waste and nutrients. So those conditions are not what is there in the body. When our target is to go as close to our body conditions, then the use of bioreactors becomes essential. So through bioreactors, we can create flow, shear flow conditions. We can like facilitate continuous exchange of waste materials and deliver nutrients. Like, so there is a continuous circulation of these two things. That is what happens in our tissue as well. We can ensure correct pH, oxygen concentrations, temperatures, a very precise manner, and so that the growth of that artificial tissue is consistent throughout the culture period. So that is why bioreactors are discussed in tissue engineering, and the more designs of bioreactors, they have been taken from chemical engineering, by the way. Okay, so they have basically taken the chemical bioreactors or the reactors, chemical reactors that they use, and modified it according to our needs. So we can add um, particular sterile conditions to the bioreactors, and then motion, fluid motions can be added of specific type, shear, electric signals can be added, magnetic fields can be added. Whatever is required to supplement the growth of the artificial tissue, we can do that using bioreactors. And commercially also, many new bioreactor types are coming up, but whatever I have seen so far, it is usually the researchers who are developing their small scale reactors in their lab and giving proof of concepts, means showing that it is possible. This way of culturing is possible. Like there is not So there is so much research to do in this area of bioreactors for tissue engineering. We still don't know many things. Okay, so yeah, um, I hope you are able to, recapitulate what you have seen for week 6 content. If you have any queries regarding whatever I said or whatever professor said during his lectures, feel free to ask. I'll give you a second to think over it and then we'll begin with the questions. Yeah. Okay. So no questions. We'll move on. So those of you who have joined in the middle of the discussion, as I was saying in the beginning of the session, that the reason for the time change is because I have moved to United States, I guess 3 days back, and because of time difference, it is around 10 hours, 10.5 hours delayed. Okay, we had just started Saturday morning, so because of that, I had to change the timing. Okay, so I hope that's not a very big change in your schedule, and the timing will be 6 to 8 for the coming 2 weeks as well. And I one more thing to inform is that I will not be able to take the extra session that I thought I would be able to take towards the end. So our last class will be on 14th September, where I'm supposed to discuss the week 8 content. But if you are studying, if you are catching up with the content, and means if you have queries on any of the lectures that is like not necessarily be that week's content, you can ask me during this 2 3 weeks time. Okay, but before your exams, if you have registered, be prepared, and take my help as much as you can. All right. But after the session as well, after this discussion sessions is over, you can always post in the discussion forum. I'm sure I will, or the other TAs or the professor will have a look at it and help you out. But don't wait for that. Don't wait for too long to get your queries solved. Okay. So now we will start with the questions. I hope this time, like last week, many of you were not there, so there was least participation in the question answering, but I hope you will participate today. Anyway, I'm here to help you. Okay. So the first question: tissue dynamics is regulated by which of these options? A. Cellular fate processes. B. Cellular signaling. C. Cellular metabolism. D. Cellular respiration. Which of these processes regulate tissue dynamics? Anyone? We just discussed. So it's a relatively very easy question. Tissue dynamics is regulated by A. Cellular fate processes. B. Cellular signaling. C. Cellular metabolism. D. Cellular respiration. Speak up if you think you know the answer. So yeah, the correct answer for this question is option A. Cellular fate processes regulate tissue dynamics. Okay. I already discussed this in the previous slide, so I'll move on. But if you have a query, let me know in the coming few slides. Okay. But I'll move on to the next question for now. In which phase of apoptosis cell commits suicide? So this question is based on cellular apoptosis. Cellular apoptosis. And it is also popularly known as programmed cell death. Okay. So there are three phases actually: the induction phase, the effector phase, and the degradation phase. And this happens in sequence, like one happens over after the another. And this process is very controlled. It doesn't cause a mess in the tissue environment. And yeah, I will discuss further about these processes and all. But if you have seen the lectures, please give it an attempt to answer this question first. Then I'll go into details of these steps that are involved in this process. Okay. So again, I'm repeating the question. The question is, in which phase of apoptosis cell commits suicide? Induction phase, effector phase, degradation phase, and all of the above.
Degradation phase.
Okay, okay. So it is actually the correct answer is actually effector phase. So let me come to why. So the first phase of cell apoptosis is induction phase. This is the phase when the signal reaches the cell saying it that it has to undergo apoptosis. Okay, so the process starts from here, start of apoptosis, because of some uh, signal. This signal is usually called the death signal, and it can come either due to, say, chemical or irradiation of non-ionizing radiation. So some harmful rays, harmful light rays can cause cell death, or it can act as a signal to cause cell death. Harmful electromagnetic rays, or removal of survival signals. So there are receptors, cell receptors that are bound to some ligands, and these receptor ligand interaction acts as a survival signal. Means telling the cell, yes, you have to stay alive. So if you remove those survival signals, then it means, otherwise means that the cell will undergo cell death. So removal of survival signals. Okay, so these are death signals, types of death signals that actually causes the induction phase. This is the first phase. Now, once the induction phase is received, starts the effector phase. This is when the executioners of the cell apoptosis, they are activated, and these are proteases actually, which comes under, which are called as caspases. Cas Phases. So there are various caspases, caspase 1 to 9. So each caspase has a role to play, but these caspase proteins are the proteins that are now activated through the arrival of a death signal, and these caspases will actually be the one to kill the cell. All right, means it will like cause the cell death. So yeah, that is why the answer for this question is the effector phase. It is the effector phase where actually cell stops functioning. It is no longer alive because of the role of caspases. And third is a degradation phase. Now, as I told you, cell apoptosis is a very clean procedure. Like it does not leave behind any mess. It is because of this phase, degradation phase. Once the caspases have done the job, what is left behind of the cell? It is basically the bilayer membrane and all the organelles and associated, right? So here what happens? This entire structure becomes fragmented, and within each fragment holds small fraction of the cellular content. So it will hold some cytokine or the cytoplasmic contents, nucleus content. So it is very much fragmented, and the enzymes, lysozyme enzymes can now easily act on degrading these small fragments. That is the degradation phase. So at the end of apoptosis, you will see no trace of the cell that was there previously. So it leaves, so it makes the job for the tissue easier. So yeah, that is why the question answer to this question is B. I hope Briefly, I could explain to you the cellular apoptosis process. If you'd like to read more, there is the lecture. Okay, go back and see the lectures. Sir has very nicely discussed it. Okay, yeah. Now, this is the next question. The question states, in which phase of wound healing granulation tissue is formed? A. Maturation. B. Proliferation. C. Inflammation. D. Hemostasis. So this is talking about the one of the dynamic state of tissue, that is tissue repair process. So under tissue repair process, or it is also called the wound healing process, we have seen there are various phases involved. So if I were to order these phases according to the um, time scale it happens on, so the first phase is basically the hemostasis. Okay, first phase. Second phase that will come immediately after hemostasis is inflammation. Then proliferation phase, and last is maturation phase. I'll discuss the details of each phase a bit later, but first you guys try to recall what you have already studied in the lectures and yeah, give an attempt to answering this question. Again, I'm repeating. In which phase of wound healing granulation tissue is formed? A. Maturation phase. B. Proliferation phase. C. Inflammation phase. And D. Hemostasis phase.
Proliferation.
Yes, you're correct. You're absolutely correct. So, those of you who didn't understand why this option is correct, let me go into the detail of it. So, first phase that I told you is the hemostasis, or it is also called the blood clotting phase. Here, where the hemo blood is caused to stay static, hence the name hemostasis. So, whenever the tissue is injured, you will see blood flow out, and that is not good for the body. Your barrier is compromised. So, that is when the blood clotting factors, which are there in our blood, and there is also the clotting cascades, these all processes starts activates immediately. It activates with the help of the cell type platelet. Okay, so upon activation of the blood clotting processes, you will see a temporary barrier is formed, that is called blood clot. That is temporary in nature. It does not mean the tissue is healed. It just temporarily stops the blood flow. So it happens due to formation of fibrin clot. So there is fibrinogen component in the ECM. When it in presence of the enzyme thrombin, it forms a gel that is called fibrin gel. We also use it for tissue engineering artificially as well, but it is also there in our body. So this gel basically covers the wounded area. So thus you will see a blood clot form. Now, when the blood clot is formed, your barrier is like intact. Whatever in the meantime, whatever bacteria or foreign objects or dust particles or whatever foreign objects have entered into your body through that injured site, now those cannot stay in our body. So that is when the process of inflammation happens, where the um, immune cells like macrophages, monocytes, okay, come into picture. What they do, they immediately go to the site and target whatever foreign particles have entered at the tissue wounded site, and they try to kill it. So a very sign of this process has started is very familiar. You guys must have been very familiar with inflammation in day-to-day life. You suppose an ant bites you and there's an irritation and you itch on it. After some time you will see swelling, okay, and also redness, and then it will become a bit warm. So these are The signs of inflammation means the macrophages or the monocytes, they are at work. They are the blood flow has increased at the site, which is in turn causing more and more infiltration of these immune cells. Also, due to increase in the blood flow, the blood vessels at this site, they expand. The local temperature of the site increases due to blood flow. So all these things are happening. Okay. Now, as the inflammation process is going on, let's suppose it is acute inflammation, means it's a very short lived, but if the damage is too deep, it can go for a very long time. All right. That is when the intervention is required from medicine to help you out. But let's suppose it is acute inflammation, then starts the tissue remodeling, or you can say then starts the cell proliferation process that is going to supply new cells to take care of the cells which is already compromised, if that makes sense. So that is the proliferation phase. So if suppose it is skin, you have lost some cells like fibroblast, keratinocytes, some endothelial cells. These cells are lost because it has been cut. So in order to replenish those cells, the proliferation phase begins. And in this process, a very native tissue is formed. That is called granulation tissue. That is a signature of this stage. If if any of you have done, I think most of us have done, especially when we were children, when we had a cut, the black substance that was blood clot, and we were always curious to, itch it out and to scratch it out, right? So when we used to do that, we used to see that the color of that region is not matching your surrounding area. It is not matching, like it is not normal. It is slightly pinkish, and it is a very translucent type of, that is the mark of granulation tissue is formed. Here, deposition of collagen, the fibroblast cells. So because of fibroblast cells, collagen starts depositing. Then fibroblast also changes its transdifferentiates into a cell type called myofibroblast that tries to contract or pull the both ends of the wounds together. New blood vessels starts to form. That's why it is pink, right? New capillaries and all which are teared due to cut, those try to form. So that is the mark of proliferation phase in progress. Now, once that is completed, uh, maturation phase starts. It is also called remodeling. This is the phase that tries to make the tissue structure now as close to as possible to what it was previous the wound. All right, it tries to restores the original structure, that is remodeling. It is not successful in adults. It is, we are not able to completely capture the normal tissue structure. That is why we get scars. So formation of scars is the sign of this phase, and this scar becomes milder and milder. So initially it is very prominent, then it becomes milder and milder over the period. This means that this remodeling phase goes on. It can go on for month to years. All right. So this is what is wound healing, and um, people are usually taught the skin wound healing because this is what usually very familiar to us. We have seen it already so many times in our life, but the same process goes on inside our body as well when our organ tissue inside the body is also damaged. Yeah. So these are the four steps, and briefly I try to summarize them for you. If you have any queries, for a second, um, let me know. I'll wait for a second. Okay. Then I'll move on. Yes, any queries from anyone? All right, moving on. So this question states or asks you, fetal wound healing is scarless because option A, wound closure happens due to rapid assembly of actin purse strings. B, basket weave form of arrangement of collagen fibers. C, both A and B. D, neither A nor B. So which of these options explains why fetal wound healing is scarless? Take a few seconds, and if you know the answer, let me know. Anyone? A slight guess would work. Don't worry if you're, yeah, someone was unmuting. Go ahead.
I'm not sure, but is it A?
All right, yeah. So you're partly correct. Okay. The correct answer is C. Both option A and B are correct. That is actually responsible for making the fetal wound healing scarless. So, what happening means these two process how it is different from the adult wound healing. So, as I told you, in adults, the scars are formed, right? It is mainly because first point there is a role of a cell called myofibroblast. So these are basically cells containing both the characteristics of a muscle and a fibroblast. So they are important for pulling. So if this is the wound edge, so this myofibroblast they try to like pull together both the ends of the wound and seal it. So that is how contraction happens in adults. Whereas in fetuses or when we are very little babies, like within a year or two, there what happens, the wound healing process or the closure of the wound does not happens like this yet. So uh, there is assembly of actin filaments. Actin filaments are there inside our cells. They are the cytoskeletal elements of the cells. So they form a string around, like they form a string around the wound. So this is the wound. They try to form a string around it, and like a purse, it tries to like pull it. So as a result, you will see no wound, no scar. Okay, means no scar forming. Okay. Now, during the remodeling step, I told you that the collagen fibers they try to reorganize themselves. They try to mimic the original state as much as possible. But whereas in fetuses, the arrangement of collagen fibers is slightly different than the adults. In adults, it is the fibrils form. Okay, one over the other, there is stack of fibers. Whereas in fetuses, the arrangement of collagen fibers is like a basket weave. So that also contributes to a scarless healing. So yeah, I hope you're able to understand now why both option A and B is correct. If you have any queries, let me know. I cannot stress it enough. This is the now. On to the next question. Lymphocytes are formed during the hematopoiesis process when the hematopoietic stem cells commit to underscore blank lineage. So this is talking about cell differentiation. So we have now moved on to cellular differentiation, topic of the lectures. So there is a process called hematopoiesis. It is the process that will give rise to various blood cells, okay, from stem cells. So the stem cells are the hematopoietic stem cells and they reside in the bone marrow. So stem cells, they can commit along multiple lineages. So this hematopoietic stem cells, they can commit along multiple lineages. So if this is HSCs, in short, it can undergo differentiation either in along myeloid lineage, lymphoid lineage. Under myeloid lineage, there is erythrocytes. Sorry, let me correct. The process, I don't recall the exact name, but it is something ends with SIS. Okay. So this is erythrocytes. So once the stem cell uh, like decides under which lineage to go, it will give rise to the fully differentiated cells that comes under each of these lineages. So that is what being asked here. You're asked here to tell lymphocytes belongs to which lineage, myeloid or lymphoid.
Ma'am, it is the lymphoid lineage.
Yes, you're correct. So the names are very similar to each other. So under lymphoid lineage, apart from lymphocytes, there are other cells as well, like natural killer cells and some special kind of dendrites that also forms. And lymphocytes, the B lymphocytes, uh, B cells, T cells. So these are formed when the hematopoietic stem cells commit along lymphoid lineage. Whereas under myeloid lineage, apart from erythrocytes, there is other type of cells as well, like granulocytes or granulocytes. Okay, monocytes and all. So these are all comes under myeloid lineage. For detailed, like recalling the detailed chart of hematopoiesis, the lectures have it. Okay, so go through the lectures, you will find the chart. Now this is the next question. The surface marker CD34 is unique to which type of stem cell? Hematopoietic stem cell or mesenchymal stem cell? In fact, this is such an important fact that people use it in their research to identify the different stem cell populations. Okay. Yes, go ahead. If you're willing to answer, go ahead. Option A. Yes, you're correct. So CD34 is unique only to hematopoietic stem cells. It is not expressed in mesenchymal stem cells. Now, can anyone try to give a situation where this information would be useful for recognizing the hematopoietic stem cells? You guys have studied about cell isolation, right? How do we isolate cells? Do you think we can use this information there? Yes or no?
Yes, ma'am. Because it is a surface marker, it will help to isolate.
And which technique would you use to isolate? We'll use flow cytometry, okay, with FACS, also known as fluorescence activated cell sorter. So in that way, we can isolate only the hematopoietic stem cells because they have CD34 marker. All right. Yeah. Answer the next question. Mesenchymal stem cells differentiate into the following cell types except A. Osteoblast, B. chondrocytes, C. myocytes, D. fibroblast. Which of these differentiated cells does not arise from mesenchymal stem cells?
And is it D?
Is it D or B? What did you say? Can you repeat?
D, ma'am.
B.
D.
Chondrocytes.
No, ma'am. Fibroblast.
Fibroblast. Yes. Yes, you're correct. Fibroblasts, they do not arise from mesenchymal stem cells. Okay. So these are all the cell types of connective tissue. Okay. Like bone, cartilage, and muscles. So it will help you remember in that way that mesenchymal stem cells help in the formation of connective tissues. So yeah, that's a good way to remember this fact. So next question. Which of the following ECM protein has RGD domain? RGD, you are guys are already familiar. This is a small peptide that helps in cell adhesion. So these proteins which are listed, the collagen, thrombospondin, and actin, and D is all of the above. So all the three proteins that are listed here are the ECM proteins. So which of this protein has RGD domain means helps cell adhesion? What does
All of the
RGD?
above.
Yes, yes, you're correct. So you're correct by the way. So but what does RGD stands for? Anyone? The three amino acids. The name of the three amino acids. Anyone?
Arginine, glycine, aspartame.
You're correct. So R basically these are the single letter short forms of these three amino acids, which one of you just stated, which is correct. Okay. So next question. Dash aid in the process of cell adhesion. A. Cell receptors. B. Transmembrane proteins. C. Proteases. D. Cytokinins.
Cell receptors.
Yes, you're correct. It's cell receptors. So cell receptors are actually the transmembrane proteins. So we have to be very specific in choosing the correct option. It's self-explanatory. It is not the proteases and it is not the cytokines. So it is cell receptors that help in cell adhesion process. At very close distances, which type of physical force is dominant for cell adhesion to a surface? Again, I'm repeating the question. At very close distances, the close distance may be the distance between the cell and the surface. That is the distance I'm talking about. So, at very close distances, which type of physical force is dominant? Which type of physical force is dominant for cell adhesion? So, before you guys start thinking over this question, let me quickly ask one question related to this question that I'm showing you. Is the receptor ligand interaction a physical force or a covalent force, means a chemical force? Ligand receptor binding, is it a physical phenomena or a chemical phenomena?
Physical.
Yes. Why? Why did you come to a conclusion that it is physical? Can you explain us?
Since they have hydrogen bonding, the non-covalent forces.
So it is not just hydrogen bonding that plays into this phenomena. It is also the configuration of the ligand and the configuration of the binding site. So due to conformational changes also, the binding happens. But it is reversible. Means one the ligand can move out of the binding site. As long as the ligand is attached to the binding site of the receptor, the signal is active and it can easily leave, and then the signal becomes inactive. So the process is reversible in nature. It is not permanent, like there's no covalent bonds forming, and that's why you don't see a signal persistent, okay, over a period of time. Means it is changing because it is a physical forces are involved in keeping the ligand and receptor intact. Okay, now first we cleared that. Now we'll come to this question again. I'm reading this question again. At very close distances, which type of physical force is dominant for cell adhesion to a surface? A. Electrostatic forces. B. Receptor ligand binding. C. Van der Waals forces. D. Steric repulsion.
Van der Waals force.
Okay. So actually, you're not correct. At very close distances, it is the receptor ligand binding that is dominant. So let me explain you. Sorry. Um, I think I marked it incorrectly, but the answer is receptor ligand binding. So this is the graph that shows why I'm correct. This is the graph where the y-axis is interaction potential and the x-axis is separation distance. As we move further away from interaction potential, this is the lowest distance or the closest distance, means adjacent the cells or the surfaces distance is the smallest, and as we move away distance is increasing. So let's start from here. As the distance is increasing, so at this point this is the closest distance that is possible, and that is the point when the interaction potential is negative, means an attractive force is possible. Now, as we are moving farther away, means as the cells are separating or as the cell is separating away from the substrate, then comes the van der Waals interaction that tries to uh, like pull the cell towards the surface or tries to keep that distance cells together. But this is a very weak force, and it's a very basically depends on the polarity of the surfaces. So yeah, it's relatively very weak as compared to the receptor ligand binding. And as we are moving farther and farther away, then comes electrostatic forces and steric repulsions. That is basically repulsing. That is now going to play a role, now keeping the cell as far away as possible from the surface. So you see the line is going towards the repulsion area. So if we the interaction potential has two extremes here, as you are able to see, attraction and repulsion. So as they are moving farther and farther away, the repulsive forces are dominant now. So the electrostatic forces, that is based basically to do the surface charge, they are now acting to keep the surfaces apart, cell surface and the substrate surface. Then the steric repulsion basically means the configuration, all right. It doesn't fit, basically the one doesn't fit on the other. So that is also there. So these forces now become dominant when this two surfaces are apart. But when the cells are very close to each other, then the receptor ligand force is dominant, and that is the distance required for any cellular communication to happen or any type of cellular fate processes to begin. Okay, yeah. So I have marked it incorrectly, ignored this red box. The correct answer is receptor ligand binding. Okay. This is the next question. On the similar topic, cell adhesion. The dissociation constants Kd for fibrinogen alpha 2b beta 3 binding. Okay, so this is the ligand and this is the receptor. This is one type of ligand receptor binding. For that, the dissociation constant is given. And there is another ligand receptor binding, that is biotin and avidin. So what are the dissociation constants? For the first one, it is 1.4 tens to power minus 7 molar. And for the second one, it is 10 raised to the power minus 15 molar. Now you have to tell me, based on these values of dissociation constant, which type of association is the strongest one. Is it the fibrinogen alpha 2b beta 3 binding, or is it biotin avidin binding? Based on the values of the decision constant that is given, you have to make a judgment. Okay, so let me give you some context to help you out here. As you have already answered, the receptor ligand binding is reversible in nature. So if this is receptor represented by R, this is ligand, then this is the receptor ligand complex that is formed. It is reversible. It can goes either way. And there's a constant called dissociation constant, Kd, that basically tells you how much the bond is eager to dissociate. And there's another constant called affinity constant, means how much these receptor ligands are keen to associate with each other. So these are opposite. So Kd is inversely proportional to Ka. All right. We represent Kd So if we go in the opposite direction, that is where this KD will come. And if we go in the forward direction, that is where KA will come. So if you were to write the expression of KD, it will be um, R, concentration of L divided by RL. And other way, if you were to write the expression of Ka, it will be RL concentration by R concentration of L. So what is the physical significance of these constants? If Ka is high, that means there is a more tendency to form this receptor ligand complex. The tendency is high. If Kd is high, then this particular complex will have a very high tendency to dissociate, right? So you will see more concentrations of individual uh, components, like there will be more concentrations of non-bind or unbinded receptor and free ligands. So that is what Kd and Ka means. Now, if you have understood, give it an attempt. I'll reread the question for you. The dissociation constants for fibrinogen, alpha 2b beta 3 binding, and biotin avidin binding are 1.4 in 10 to the power minus 7 molar and 10 to the power minus 15 molar respectively. Which of the above mentioned ligand receptor binding interaction is stronger? Option A, fibrinogen alpha 2b beta 3 binding, or B, biotin avidin binding. If anyone is ready, unmute yourself and you can answer. Yeah.
Ma'am, option D.
Yes, yes, you're correct. How did you come to that conclusion? Can you share? Yes. Share with the rest of us. How did you come to this conclusion that option B is correct?
Ma'am, if the dissociation constant is less, then the ligand receptor binding interaction will be stronger, as you've already mentioned here. So if the KD value is too high, then it will easily dissociate, so the binding won't be stronger.
Yes, yes, that's correct. So actually, a fun fact, this decision constant for biotin avidin binding, the value is actually closer to the decision constant of a covalent bond. So this biotin avidin binding is almost as strong as a covalent bond. Yeah, and that is why it is very, very widely used for various assays where they try to use this interaction in order to tag some fluorescent dyes or some enzymes and then perform some reactions. So yeah, if anyone has any queries, let me know. Okay. The tensile strength F of receptor ligand bonds, it can be estimated using equation. Which equation helps you estimate the value of the tensile strength of receptor ligand bonds? So F is the uh, letter that is used to denote the tensile strength. So option A, F is equals to delta G naught by small delta. So delta G naught here means the Gibbs free energy. Here delta means the bond length. Option B is F is equals to delta by delta G naught. Option C is F is equals to delta G naught by Ka. So Ka from the previous slide means the affinity constant. And D is F is equals to Ka by delta G naught. Which equation do you think can help you determine the tensile strength of this type of receptor ligand bond? If you remember, the equation for work is very similar. Okay, maybe it will be difficult. This question might be difficult for some of you. Let me ask you some other question related to this. If the length or the distance, if the distance between say cell A and cell B, this is the distance delta. If distance delta is high, will the uh, strength of this bond F be higher or lower? Forget the question. First, answer this, what I've just asked. If you increase the distance between cell A and cell B, that is, if you increase the value of delta, will the strength of the bond will be higher or lower?
Lower.
Lower, yes. So this means F is inversely proportional to delta. Okay, now tell me another thing. For formation of a bond, you need some energy. Okay, so here the energy that we're going to use is Gibbs free energy. But why? What is Gibbs free energy? There has to be some energy associated for a bond to form, right? A stable bond will be formed when this energy is like supports, right? Thermodynamically is favorable. So what is then Gibbs free energy? Why not kinetic energy or potential energy or any other energy you have already learned? Why the role of Gibbs free energy will be important here? What do you understand when someone tells you this is the value of Gibbs free energy?
Ma'am, is it related to equilibrium? I can't remember correctly, but I think it is related to that.
So you're partially there. But let me tell you what Sir has explained. Okay, Sir has explained it very nicely, where in a very general term that applies here. So Gibbs free energy refers to the energy that is not being used to do a work. Okay, so that energy is not consumed to maybe move some things from one place to another. So that is what is Gibbs free energy. So when you say equilibrium, we usually study it when we are talking about chemical equilibrium in reactions. Their work is not happening, but still some energy is being used, right, for facilitating that particular reaction and bring it to an equilibrium state. So that is where Gibbs free energy works. So when it comes to receptor ligand bonds, the formation of these receptor ligand bonds, there is no what do you say, the bond is only forming because of affinity. Means if the ligand is of correct structure, if and shape, then it can easily fit into the binding site of the receptor, right? So it is based on the affinity, the distance. So some energy that is involved that makes sure that this bond is formed without any active involvement of any external factors. That is Gibbs free energy. Okay, here that is why we are talking about Gibbs, using the Gibbs free energy for describing the strength of the bond. Okay, now from this discussion, you tell me which of these options or which of the equations given in the options, four options, will help you determine the value of tensile strength. Is it option A, B, C, or D? So you already know that force or F, sorry. Yeah, go ahead. Sorry. Some one of you was speaking. Go ahead.
A.
Yes, you're correct. It is option A. All right. So as I was saying, that this tensile strength is inversely proportional to delta bond length, and the higher the Gibbs free energy means the process is more likely to happen. Yeah. So the strength of this force will be higher. Yeah. Strength of this bond will be higher. So yeah. Which of the following acts as a mechanical link between cells? So here I'm talking about the various cell adhesion receptors involved, okay, that forms junctions, various types of junctions. So there are three types of junctions mentioned here: the tight junctions, anchoring junctions, and communicating junctions. Now, which junction type acts as a mechanical link between the cells?
Anchoring junction.
Yes, you're correct. So for those of you who didn't get it, let me show you the images of these junctions. So it will be much more clear, become much more clear to you. Okay, so I have it in the upcoming slides where I have a similar question of this type. So yeah, this question is about the similar cell adhesion junctions. It is a match the following question. Um, tight junctions what do you, if you know what the meaning of tight junctions is, it then try to answer like it matches correctly with either one, two, or three. Similarly, adherence, hemidesmosomes, they are types of anchoring junctions, and each of these are responsible for a type of linkage in the cell. First, try to answer this question, and then I'll come to the explanation part of different type of junctions and all. Okay? Yeah.
Tight junctions are impermeable barriers to fluids, and adherents are those link actin filaments of adjacent cells, and hemidesmosomes link intermediate filaments of cells.
You're correct, actually. Okay, you're correct. So, those of you who don't recall what are these junctions and how they work, I have some images that will help you. So this is a representation of how a tight junction looks like. So the proteins, there are various proteins in the membrane. They are equally contributing from the adjacent cells to create an impermeable barrier to fluids. Means nothing, almost nothing can pass through between the cells. It is very prominent. It is very important for epithelial lining, suppose. Okay, so if anything were to pass through this junction, then it has to use active passage, means the concentration have to be like not against, but towards it, means some energy has to be consumed in order to allow the passage of anything through tight junctions. Now, there are various kind of anchoring junctions. So in this image shows few of them, the desmosome, hemidesmosome, and adherens junction. So desmosomes are basically formed by a class of cell receptors called cadherins. So these are the cadherins, all right, representation of cadherins. They these cadherin receptors, they link the intermediate filaments, link the intermediate filaments that are part cytoskeletal of the cell, and in this way they mechanically link the adjacent cells. Whereas adherent junctions, they have a different kind of receptors involved. They are called beta catenin and all. So these are the representation of those receptors. Now they are involved in connecting the actin filaments. So these are the representation of the actin filaments. Okay, this actin filament is also a part of cytoskeleton. Now, there is another type of adherent junction formed by the hemidesmosomes. Now, how hemidesmosome is different from desmosome and adherent junction is these hemidesmosomes are responsible for anchoring the cell to substrate. Remember, here desmosome and adherent junction, they are adhering the adjacent cells through receptors, whereas hemidesmosomes uses integrins as a receptor to anchor the cell on the substrate and link the integrin receptors to intermediate filaments that are there inside the cells. Okay, so in this way, um, like through receptors, the cytoskeletal elements are linked. In this way, they're mechanically linked either to each other or to a substrate. Okay, I hope this is clear. Now there is another kind of junctions that I told you in the previous question, that is communicating junctions. These are basically porous junctions, maybe like some transmembrane proteins are involved, and there is a pore running in the middle of it, through which small ions or small molecules can pass through in and out of the cell. So they can allow either one directional passage, uniport, symport. So these kind of junctions allow intake of materials and out and repulsing them outside the cells. These are communicating junctions. These junctions also helps in secretion of various growth factor molecules and further facilitate cell communication. Now selectins are cal sorry, not okay. Selectins are chloride dependent binding proteins expressed during repair phase and involved in inhibition of neutrophil migration. Is this statement true and false? So, things to note: selectins are, is it chloride dependent first? Is it expressed during the repair phase? And are they involved in inhibition of neutrophil migration? So the answer to all these questions is one, either it is true or false. Selectin is a type of cell receptor that is involved in forming adherent junctions. Okay,
False, ma'am.
you're correct. For those of you who are not sure, this is the slide I took an image of from the lectures. So this describes the role of selectins basically. First thing, selectins are calcium dependent binding. They show calcium dependent binding. Means if the calcium ions are present, the selectin will be able to like link to the adjacent selectin receptors and form bond. So, very interesting fact, these calcium ions are divalent cations, right? So, if you use a chelating agent like EDTA salt, then you remove the calcium ions and then reverse the binding. So, yeah. Then these selectins, they are transiently expressed during the inflammation response. So they are not expressed every time or prolonged period of time. They're only expressed during inflammatory response. They are typically expressed on the surface of endothelial cells that line the artery walls. They contain a lectin domain which help recognizing specific oligosaccharides which are expressed on the surface of neutrophils. They help in migrating, they help uh, endothelial cells to bind to neutrophils, then allow the migration of the neutrophils, and eventually transmigrate through the blood vessel wall to participate in inflammatory response. Okay, so that is what is the role of selectins, and yeah, so and knowing what we know from the lectures, this option is correct, means the statement is false. All right. If you have any queries, please let me know. This is the next question. Which one of the following cell which of the following cell receptor type requires calcium to facilitate cell matrix adhesion or cell-cell adhesion? Select all that apply. Means here more than one option is correct. So these are the names of few cell receptors involved, all right, in forming adhering junctions or mechanical links. Okay, but few of these require calcium ion for facilitating adhesion, and few of them do not. So you have to select which of these require. Option A, integrin; B. Cadherins, C. Selectins, D. Ig like CAMs. Ig immunoglobulin like cell adhesion molecules. Anyone? Okay. So as I already told you, the selectins require calcium. That is, C is correct. Whereas other type of receptors like cadherins and integrins, they also require calcium. So these are called calcium dependent receptors. Whereas receptors like Ig like CAM, they do not require calcium, or they are calcium independent receptors. Okay. This is the next question. Consider the migrating cell that is shown in pink color and the brown color is the substratum. Here, which is leading edge, A or B, and it is called as 1. A is the rear region and is called as the Europodium. B is the rear region and is called the Europodium. A is the leading edge and is called the Lamellopodium. B is the leading edge and is called the Lamellopodium. Which of these option is correct? 1, 2, 3, 4.
Third option.
Yes, you're correct. What is so A is the leading edge here, is the leading edge, and B is the rear edge, a rear region, and the rear region is called Europodium, and A is called the Lamellopodium. So these two edges basically help in translation of the cell from position A to position B. Okay, one position to another. What happens during cell migration is that the leading edge or the lamellopodium, it moves few steps forward after the cell and substrate binding is removed. That is why it moves a few steps forward. As it moves forward, the cell stretches out. That causes the rear edge to pull, all right, and move forward. In this way, it has moved one position forward. Okay. The period when the cell is moving forward in a straight line without turning is known as A. Net average speed, B. Random motion, C. Hayflick limit, D. Persistence time. This question is again related to cell migration and it is asking you uh, the terminology that means that refers to the time period when the cell is only moving forward in a straight line without turning. What is that time period called?
Persistence time.
Moving on, you have to match the type of the migrating cell with its role. Okay, neutrophils, the type migrating cell that are given is neutrophils, endothelial cells, tumor cells, fibroblast, and some of the roles mentioned is metastasis, wound healing, angiogenesis, and phagocytosis of bacteria. One by one, you can address. Option A, neutrophils. What its role in the body? Why it migrates?
Phagocytosis of bacteria.
Yes. It tries to kill the foreign material that has entered, that is a bacteria, through phagocytosis. Then endothelial cells, why it migrates? For which of the purposes it will migrate?
Angiogenesis.
What about tumor cells? They are known to migrate to cause.
Metastasis.
That leaves fibroblast, which are known to migrate during their proliferative phase and cause healing of the site, wounded site. This is the next question. For neutrophils, P is the persistence time, by the way. P is the persistence time. Its value is 1 to 4 minutes for neutrophils, where S is the speed of migration. For neutrophils, it is 20 micrometer per minute. Whereas for endothelial cells, uh, P is 300 minutes and S is zero point 0.4 micrometer per minute. So, which cell population has a higher motility than neutrophils or endothelial cells? So, as we have already seen, the meaning of persistence time, it means it will, uh, it is a time period for which the cell is moving forward in a straight line without turning. That is persistence time. And the speed is obvious how fast it is moving through that distance. If you were to given these values of S and P, can you estimate, like, can you guess which cell population is more motile? Is it neutrophil or endothelial cells?
Neutrophils.
Yes, you're correct. Why neutrophils? Because it is changing its direction very frequently than endothelial cells. Like every minute, the cell is turning, right? And the speed, if you see, it is much higher at which the speed at which it is moving, it is much higher than the endothelial cells. And it is obvious given that neutrophils are involved in phagocytosis process, means it is always on move and searching for any foreign object that has entered and try to kill it. Whereas for endothelial cells, it is not the obvious thing for to migrate. Their function is to form a stable blood vessel wall and allow a movement of circulation of the blood. So yeah, that is why that in this way you can estimate which cell population is more motile just by experimentally estimating the values of persistence time and speed. Which of the following signaling molecule control the generation of tissue formation? Sorry, I think it was not visible earlier. Here is the question again for you. Let me read it. Which of the following signaling molecule control generation of tissue formation. Option A, mitogen. B, morphogen.
Morphogens.
Yes, you're correct. It is the morphogens which help in or which add the signaling molecules which help in generation of tissue. That is during development of an organism. These morphogens are highly active, whereas mitogens are responsible for signaling cells to divide when it is needed, like undergo mitosis. That is when the mitogens come into picture. This is the last question for this session. Which conditioning types are applicable for cartilage cell culture in a bioreactor? So, what type of stimuli, what type of stimuli would you give for culturing artificial cartilage in a bioreactor system? So, these are different type of stimuli that you can give through a bioreactor to supplement the growth of a artificial cartilage. First, option A, compression, B, electrical, C, shear, and D, magnetic. So, which stimuli is more applicable or more appropriate for culturing cartilage artificially in a bioreactor? So, remember, bioreactors are meant to mimic the in vivo environment that the cells or the tissue is in. All right. So, we are trying to mimic that. So, you have to remember under what conditions the cartilage is there in our body, what kind of forces are always persistent at the cartilage. You know where the cartilage is and which body parts cartilage is found, you know, a very common joint, in a very common joint, cartilage is formed. Sorry, cartilage is found. Anyone? Yes.
Joints.
Yes. Which joint? Which joint? Very
Um,
commonly discussed, actually. We have discussed a procedure called autologous chondrocyte transplantation. It happens at which joint?
hip joint.
Actually, it happens at the knee joint. That procedure happens at the knee joint. So if you imagine your knee, under which forces it is always in, which kind of forces it is always in? When you walk, when you sleep, when you sit, what type of forces are being applied to those joints? These bones are lined with the cartilage and they actually help in easy movement of the knee. So similar forces will be needed if you want to mimic the function of the actual cartilage in vitro.
Compression.
Yes, compression. What else? Actually, here more than one is correct. Compression is one. What else?
Shear forces.
Yeah, yes. So these two forces are dominant in your body, and that the cartilage in your body experience these two forces dominantly. So if you want to successfully culture artificial cartilage tissue in vitro in a bioreactor, it will be helpful if you these give if you give these kind of forces to it. Okay, so yeah, that the questions, I'm done with the questions for today. I have few 10 minutes, I think. I have finished early today. So if you have any queries, let me know regarding the lectures of this week or previous week. I'm here for you, and yeah, feel free if you have any queries. Yeah. I see people are leaving, so most probably they may not have any queries for now. Others, um, if you have any queries, I'm here for a few more minutes. We can discuss. Else, you guys can leave the meeting.
We are clear with the session. I understood everything.
Oh, that's great. Thanks for the feedback. Um, what about others? If they have a feedback to share or not, if there are any queries. Like, I hope you guys are going through the lectures week wise and attempting the assignments. If you face any trouble in understanding any assignment problem, that also we can discuss. But I cannot help you and give you answers to the existing assignment that is active. But I can discuss the assignments that is already submitted, and if you have any questions related to that as well, let me know.
Ma'am, no doubts.
Okay, that's great. Okay, then, thank you all for joining. I hope you understood everything for today's session and you enjoyed it. I hope the questions were useful to you. The recording will be available to you soon. And yeah, good night for you guys. And it is the starting day for me. Um, bye bye. Thank you all for
Bye,
joining.
ma'am. Have a good
Yeah,
day.
yeah,
Thank you,
thank you.
ma'am.
Yeah.